Welcome to our third podcast on the respiratory system. In this third and final podcast, we're going to be looking again at the hemoglobin molecule a little more closely, specifically at the ability of hemoglobin to load oxygen and unload oxygen, as well as we'll see how the blood transports carbon dioxide. The red blood cells play a role in that, but probably not what you think. And then we'll also look at possible disease states and also pH balance within the blood. So we've already seen that hemoglobin is the molecule that transports oxygen. When we dissolve oxygen in the blood with our 100 millimeters of pressure, that's our partial pressure, that's going to result in about 20 mils of oxygen per 100 mils of blood. Some of that's dissolved in plasma, but a lot more of that's going to be bound to hemoglobin. Now recall, one hemoglobin molecule consists of 4 chains, 2 alphas and 2 beta chains. Each one of those chains has a heme group that with its iron can bind a molecule of oxygen. Each red blood cell has over 280 million molecules of oxygen. So each single red blood cell can carry over 1 billion oxygen molecules. Now, when you consider the amount of red blood cells in your blood and the fact that you have 5.5 liters of blood, what is our answer typically? There's a lot of oxygen circulating in your bloodstream. So here we see an illustration of our 2 alpha chains, 2 beta chains. The red is the heme group with the iron bound in the center, and that will be the oxygen binding motif. That's the functional binding part of hemoglobin. So since hemoglobin can be bound, then we can call hemoglobin oxyhemoglobin when oxygen is bound to the iron particle, or deoxyhemoglobin after the oxygen has been unloaded to the tissues. Now we also see in small amounts and not necessarily significant amounts, we can have methemoglobin, and this is going to be a different charge state of the iron, and it doesn't really do a lot as far as oxygen transport. But we do have carboxyhemoglobin. Now, let me just caution you here, because notice, this is not carbon dioxide binding. It's carbon monoxide. And carbon monoxide binds 210 times stronger than oxygen does to that iron molecule. This is why carbon monoxide poisoning is so dangerous, and really the only way you're going to rescue someone that is suffering from carbon monoxide poisoning, if they're still alive, is to flood their system with oxygen, overwhelm the system with oxygen, and then as the carbon monoxide is coming off and on the hemoglobin, because it's going to be the net movement, we're going to be continually moving, there's going to be so much oxygen that it's going to outcompete the carbon monoxide, even though the carbon monoxide has a stronger binding. So that's the only way to rescue someone that's suffering from carbon monoxide poisoning. Now, when we talk about transport of oxygen, we can either load hemoglobin up with oxygen, and that's going to happen in the lungs. And do you remember what kind of respiration that is? That is external respiration. So oxygen loading happens during external respiration in the lungs. Now, the opposite, unloading, you're right, internal respiration, where oxygen is given off to our tissues. Now, what's going to regulate this? Well, the concentration gradient of oxygen, the pressure gradients between the hemoglobin and the environment, either the lungs or our tissues. And then we're going to see the affinity of hemoglobin for oxygen, because we can in fact alter that. And in very specific ways, it's going to be important that we do alter that affinity to either increase the unloading or decrease it. So just a refresher, here in our lungs, we have external respiration. Oxygen is being loaded into hemoglobin. Internal respiration, oxygen is being unloaded from the blood to our tissues. Now, as we describe the loading and unloading, we're going to see that we can create a dissociation curve. Based on the partial pressure of oxygen, we can look at how saturated our hemoglobin is. Now, at our 100 millimeters of mercury at atmosphere, you remember what our saturation point is? 97%. That's what we read with our pulse ox. But as we look at our curve, as we lower that pressure, we're going to saturate less. And so we're going to be developing this dissociation curve based on partial pressure and saturation points. So if we are at 97% saturated, which is normal, as our blood passes through our tissues and we experience internal respiration, only 22% of our oxygen is going to be unloaded at any one time. Now, it's just like we saw with respiration. It's like we saw with heart contractility. We have a built-in reserve. So almost 80% of our oxygen is not going to be used, so that when we do experience a need for it, maybe heavy exercise or a very sympathetic response, we can unload a lot more oxygen, especially to our skeletal muscles. And that may be upwards of 80% of our loaded oxygen may be dissociated to the tissues. So here is our normal dissociation curve. Here is our saturation of hemoglobin with oxygen. Here's our partial pressure of oxygen. And we can see normally at 100 millimeters of mercury of oxygen, that's at atmosphere, we're basically at our 97%, practically 100%. And there's a fairly broad range of partial pressures. I mean, we can go down to 80 and we're really still at almost 97%. Even as low as 40, remember, this is what it's going to be in our veins, we still have, 75+% of our oxygen loaded up. Remember, because we only unloaded 20%. That's what we see here, just that 22%. And so if we modify this dissociation curve, then we can really see a difference as far as how much oxygen or how little oxygen we give off to our tissues. Here's a short video, and hopefully this will help you increase your understanding of oxygen loading and unloading and the dissociation curve.
All cells need oxygen. It is the essential fuel which is necessary to enable cells to stay alive and to carry out their various activities. Bringing oxygen to the cells requires the uptake of oxygen from the air in the lungs, its transportation in the blood, and its delivery to cells all over the body. The first step is the taking up of oxygen by blood flowing through fine capillaries in the walls of the lungs' air sacs, or alveoli. The oxygen molecules change from their state as a gas freely circulating in the air, dissolving into a solution in the plasma within the capillaries of the alveoli. Once in the solution of the blood, 98% of this dissolved oxygen is taken up by passing red cells, leaving just 2% remaining in the physical solution unattached. Red cells are particularly well suited to transporting oxygen because they contain a special oxygen-binding protein known as hemoglobin. Each molecule of hemoglobin itself contains 4 molecules of heme, an iron-containing pigment, which binds oxygen loosely and reversibly. Hemoglobin that is fully saturated with oxygen is bright red and is called oxyhemoglobin. On the other hand, hemoglobin that is not saturated with oxygen is purplish-blue in color and is called deoxyhemoglobin. It is heme which makes it possible for the red cells to pick up oxygen dissolved in the blood, transport it combined with hemoglobin, and release it back into the blood as oxygen in solution, ready for delivery to the various cells of the body. Hemoglobin gives up its oxygen as red cells travel through capillaries in tissues where there is a low content or partial pressure of oxygen. The partial pressure of oxygen represents the level of dissolved oxygen in plasma. As oxygen is released and again is carried in solution, the partial pressure of oxygen in the capillaries becomes greater than the partial pressure of oxygen in the surrounding tissues. This causes oxygen to move out of the capillaries into the tissues and to finally reach the cells. This graph, the oxygen dissociation curve of hemoglobin, shows why hemoglobin is particularly suited to its role in transporting oxygen. The oxygen dissociation curve demonstrates the relationship between the oxygen carried in combination with hemoglobin, the O2 saturation, and the partial pressure of oxygen in the arterial blood. The sharp upstroke and the flat plateau illustrate how oxygen is released to the tissues over a wide range of conditions. Its shape means that although the partial pressure of oxygen in the blood returning from the lungs and being pumped out by the arteries may be reduced to only 50% of the normal value, say due to lung disease or high altitude, hemoglobin will still be 85% saturated with oxygen.
So now let's talk about this Bohr effect. This is where we're going to alter the affinity of oxygen and hemoglobin and therefore change this dissociation curve and either make it harder or easier for oxygen to unload from hemoglobin. So what the Bohr effect describes is basically as you decrease the pH of your blood, you are also going to decrease the affinity of oxygen for hemoglobin. So we've got a direct relationship here. Now this makes sense, right? Let's think of pH of the blood. How can we lower the pH of the blood? Well, if you increase our CO2 content, we're going to create carbonic acid. That's going to lower the pH. And when do you produce more CO2 than normal? Right, when you are undergoing a lot of high metabolic activity, heavy exercise, a sympathetic response, you're running away from something. And so if you're producing a lot of CO2, that means you're burning a lot of oxygen, and you need more oxygen to get to your tissues. So it is an intrinsic, automatic, built-in system for when you're using oxygen, which is determined by how much CO2 you produce, lowers the pH, therefore more hemoglobin can unload oxygen to your tissues. Now we can also say the same thing for temperature. The higher the temperature, the more oxygen you unload, which makes perfect sense because if you're very active, your temperature is going to go up, just like when you're active, CO2 levels are going to go up. So here's our Bohr effect. Remember, physiological pH is 7.4. So here's our normal dissociation curve. And so if we lower the pH to 7.2, do you see how we've shifted this graph over to the right? So if we shift our dissociation curve to the right, then we are going to make it easier for oxygen to come off. We have decreased the affinity. We will unload more oxygen to our tissues. Now the inverse is also true. If we increase the pH, if we make it more alkaline, our curve is shifted to the left. And this is important because a left shift is going to signify an increase in affinity. We will not unload more. In fact, we will retain more oxygen with a leftward shift of our dissociation curve. So movement to the right, lower infinity, more unloading. Move to the left, higher infinity, less unloading of oxygen to our tissues. Now, we also have a situation where, like our creatine phosphate in muscle, it was sort of a reserve of energy. Well, we kind of have a secondary system in play in red blood cells where in low oxygen conditions, just based on that low oxygen conditions, we can unload more oxygen. And it's going to be a molecule that's called 2,3-DPG, diphosphoglyceric acid, and this is only found in red blood cells. And so what's going to happen when we're in anaerobic conditions, our oxygen levels are low, 2,3-DPG is going to be producing greater amounts. And this greater amount of 2,3-DPG will lead to a higher unloading. And this is exemplified, let's say, for instance, in people that have anemia. If you cut the oxygen concentration in half and you measure the amount of unloaded oxygen, normally we're going to unload that 22%, which is around 4.5 mils of oxygen. So half of that, if someone has severe anemia, would be 2.25 mils. But in fact, when you measure the amount that's unloaded, it's 3.3. And as they saw this effect and began to backtrack and try to figure out what's causing it, they discovered 2,3-DPG as a way to increase the unloading of oxygen to tissues. So another way we can do this is at altitude. At high altitude, you have less oxygen, you're getting hypoxic. You're going to produce more 2,3-DPG. So you take off the brakes. Normally you've got the brakes on. That's what this means, inhibition. So you increase 2,3-DPG. You lower the affinity of hemoglobin and oxygen, and there's your increase in unloading of oxygen to the tissues. Now, this is going to be a negative feedback, so as you unload more oxygen, you're no longer going to sense low oxygen, and so this is going to begin to shut down. So this is our negative feedback into the system. Now, as we look at different molecules that bind oxygen, we also see that there's a different type of hemoglobin. Sure, we've got the 2 alphas and 2 betas in adult hemoglobin, but in fetal hemoglobin, we're going to have 2 alphas and 2 gamma chains. And what the 2 gamma chains confer, as you look at the graph below, here's adult hemoglobin, our dissociation curve, and here's fetal hemoglobin. Now, what direction do we have a shift? Fetal hemoglobin is shifted to the left. What does a leftward shift mean in our dissociation curve? That's right, greater affinity for oxygen than adult hemoglobin. So as maternal blood flows into the placenta and comes into close proximity with fetal blood, now again, in the placenta, mom's blood is rich in oxygen, it's arterial blood. Fetal blood is returning from the fetal body, so it's low in blood, so you've got a concentration gradient 1. That's going to assist in getting oxygen from mom's blood to the baby's blood. But now you also basically have an oxygen magnet, and fetal hemoglobin is going to pull oxygen off of adult hemoglobin and make that unloading even more efficient from mom to the baby. Myoglobin is another oxygen-binding molecule, and when we look at the myoglobin dissociation curve, we're going to see that myoglobin has the strongest affinity for oxygen of any molecules. And it's said that myoglobin has an instantaneous binding tenacity. I love that word, tenacity. It really speaks to what's happening. Myoglobin is absolutely going to rip oxygen off of any molecule it comes in contact with. So if myoglobin has a higher affinity relative to adult hemoglobin, what direction would you expect the dissociation curve to be shifted? To the right or to the left? If you said left, you're absolutely right. And myoglobin is almost pegged against the graph. It's almost on the axis. It is such a dramatic increase. It's almost as far left as you can get. And so oxygen circulating through the blood in cardiac and skeletal muscle, you bet that oxygen is going to be ripped off the hemoglobin, stored in myoglobin in the muscle until you deplete the oxygen as you're going through aerobic respiration, and then myoglobin will give off the oxygen to the aerobic system to produce ATP. Now, myoglobin is kind of interesting. There's all the alligator shows on TV now, Swamp Men and whatnot. Well, in reptiles, they can hold their breath a really long time because they have such a high concentration of myoglobin in their muscle cells, and not to mention what they can do with their metabolism. And scientists have really been interested in these effects, and so they genetically engineered mice that don't produce myoglobin. Now, what could you imagine a mouse would look like that could not produce myoglobin? Do you think they would be viable? Do you think they would survive very long? What do you think? What's your prediction, young scientist? Well, these mice show no obvious defects. They seem perfectly normal. However, I would bet money if you put a myoglobin mutant mouse beside a myoglobin normal mouse, and then you released a cat, I bet I know which one would be lunch for that cat. I think that's where you will see a difference in how they handle sympathetic responses. Now, there was a question earlier, I believe it was last week, where we talked about AS and various genes. Well, genetically speaking, we can have defects in hemoglobin. And so in these populations, the alpha and beta chains, we can also have a hemoglobin S. And this is in sickle cell anemia. Hemoglobin S is going to cause the hemoglobin to be much more rigid. It's going to cause it to crystallize in the red blood cell, giving it a really angular shape. And this is all due to one amino acid difference. And that's the only difference between hemoglobin S and the alpha chain. Instead of a valine, you're going to have a glutamic acid at amino acid 6 in that position. And, it's not just that it changes the shape, but because the shape affects the rigidity of the cell, they're not as elastic. When you try to squeeze through red blood cells, you can have clotting, which could lead to strokes, heart attacks, embolisms. It's very damaging. And so the typical life expectancy of individuals that have sickle cell anemia are in their early to late 40s, males and females respectively. And so here on the left, we see our nice round red blood cells. And then on the right, you can see this sickle shape, this angular aspect of the cells that contain the hemoglobin S, this crystallized form of hemoglobin. Now, this really first began in populations of sub-Saharan Africa. So African American populations here in the U.S. are the ones that deal with this the most. Now, you would think that over the generations, detrimental diseases would not persist in the population, with natural selection. But when we see what sickle cell anemia actually did in the early beginnings, we see that because you change the shape of the red blood cell, now that red blood cell is not a great host tissue for the malaria parasite. And so these people in sub-Saharan Africa, instead of dying in their childhood years from malaria, they were protected from that and they lived until their 40s. So it was actually a survival advantage to carry the sickle cell gene in these populations. Well, we're not really all that threatened here in the U.S. with malaria, and that's why the gene still persists in our population today, because certainly early to late 40s, you survive long enough to pass your genes on to the next generation. Now, another group of people suffer from a disease called thalassemia. Thalassemia is not a problem of the wrong kind or a mutated hemoglobin. In this case, thalassemia results in an underproduction of one or both of the hemoglobin chains, either alpha or beta. And so this is a reduced rate syndrome, not a mutation syndrome. And it's going to result phenotypically in red blood cells that instead of having that nice open round jelly donut look, they're going to have bands of hemoglobin, bands of this red material going across that clear region. And so for that fact, they call them bridge cells because it looks like a bridge of tissue, or nizocytes. And that's characteristic of people with thalassemia. See, this is not uncharacteristic of what you might see with a normal red blood cell, right? You got your tissue on the edge, you've got the clear center. But when you start to see these lines going right down the middle, or even portions of tissue here, those are in fact your bridge cells and nizocytes and indicate this patient has thalassemia. Now, at this point, we can't tell if it's alpha or beta. You can't just tell that phenotypically looking at peripheral blood smear. But we do know from those that we are talking about a quantitative problem, one of the functional genes. Now, thalassemia and its inheritance, it's going to be a recessive disorder. So you must carry both alleles to actually express the phenotype of thalassemia. So in that case, both of your parents either have to have thalassemia or be carriers for thalassemia before you then have a 25% chance of being affected and expressing that phenotype. So if we mutate the alpha chain, alpha thalassemia, beta thalassemia, that is going to be a mutation of the beta chain of hemoglobin. Now, thalassemia as a term comes from the Greek word thalassa, which means sea, and hema for blood, because this is very prevalent among people in the Greek islands, parts of Italy, so people in the Mediterranean that are around that sea, and that's where we get the term thalassemia. Now let's shift our attention to carbon dioxide, just for the last portion of our time here. Now, carbon dioxide, when you're first thinking about its transport, it would make sense logically in the mind of a middle schooler or elementary school child that, okay, we transport oxygen. When the oxygen comes off, the carbon dioxide comes on because we're going back to the lung, right? Makes sense. Well, when you look at the transport of carbon dioxide, only about 20% of the transported carbon dioxide is going to be bound to hemoglobin. And we don't call it carboxyhemoglobin. Remember, that's with our carbon monoxide. Carbamahemoglobin, that is when carbon dioxide is bound to hemoglobin. Now, some of the CO2 is going to remain dissolved in our plasma, about 10%. That's where it begins. It dissolves out of the tissue by diffusion, dissolves into the plasma. But then the bulk of our carbon dioxide is going to be processed in the red blood cell, and it's going to be put into the plasma as the bicarbonate ion, and that's its chief transport form to get it back to the lungs. So how does a red blood cell process carbon dioxide? Well, it's an enzyme, and we'll see this many, many times. Carbonic anhydrase. This is an enzyme that facilitates the formation of carbonic acid. CO2 in water makes carbonic acid. This enzyme is going to be reversible. So the path of this enzyme is going to be determined by the concentration of carbon dioxide. So high carbon dioxide is going to favor the formation of carbonic acid. Low carbon dioxide is going to favor the conversion of carbonic acid back into CO2. Can you see where we're going with this process? I hope you can. All right, so high CO2 levels in our capillaries, internal respiration. So CO2, based on that carbonic acid in the red blood cells, is going to lead to the formation intracellularly of carbonic acid, which quickly is going to dissociate into the bicarbonate ion and hydrogen ion. So here we have an internal respiration, our tissues giving off CO2 by diffusion. Following that concentration gradient, it dissolves in the plasma. 10% stays. 20% of that dissolved CO2 is incorporated and bound to hemoglobin. But here's the remaining 70%, CO2, water, carbonic acid. Carbonic anhydrase is the enzyme that facilitates that. Go ahead and write Ca and put that in this little area. This quickly breaks down into hydrogen ions and our bicarbonate ion. Now, those hydrogen ions, by and large, many of those are going to combine with the hemoglobin in this cell to change the ionization state of the iron. But the bicarbonate ion is going to be transported out of the red blood cell. Now, in the past, when we saw movement of ions out, especially when they're charged, that's offset by molecules moving in to balance that. Remember our sodium, potassium that we started at the very beginning of the semester? So we're going to balance this outward move, or this outward shift, remember that word, of negative molecules by bringing in another negative molecule so we don't change the membrane potential drastically. And that ion is going to be chloride. So as our bicarbonate is formed and diffuses out, and again we mentioned that the hydrogen ion is going to bind to hemoglobin, the chloride ion moves in. And that's going to maintain our membrane potential, and it's going to shift the bicarbonate out, and it's going to be transported in the plasma back to the lungs. Now, we talked about how CO2 increases the pH because of the carbonic acid, and it's going to increase the oxygen unloading. Well, likewise, the more oxygen you unload, the more you're going to improve carbon dioxide transport and the formation of carbonic acid. So each one, almost like a seesaw, each one is going to enhance the other. And it makes perfect sense. Because the more oxygen you unload, the more CO2 you have to deal with, and you have to transport it effectively. So here's our illustration of our chloride shift. Bicarbonate out, finishing off that 70% of our CO2 transport, and the chloride ion coming back in. So this picture, this is our picture of internal respiration as it relates to the transport of CO2. Now remember, I said that this reaction, facilitated by carbonic anhydrase, it's going to be driven largely by the concentration of CO2. And at the levels of our tissues, that CO2 is high. But what's going to happen now when we transport all of this back to the lungs, and now CO2 in the environment is lower than what we have inside the cell and in the plasma, which is going to be high bicarbonate. That's right. The reaction is going to be driven in reverse. And that's the key word, because the first thing that has to happen is the bicarbonate ion has to come back into the red blood cell to be processed. And how are we going to balance off that movement of the bicarbonate ion? Shifting chloride back out. And since it's the opposite of what we see here, we call that the reverse chloride shift. And this happens in external respiration. So hydrogen ion that we produced and bound to hemoglobin, that's released. We're going to have our bicarbonate come back into the cell, combine with those hydrogens to form carbonic acid. Now that we have low CO2 levels, carbonic acid is going to be acted upon by carbonic anhydrase to form CO2 and water. Chloride moves out to balance that shift of bicarb in. Now CO2 is free to diffuse following its concentration gradient out of the red blood cell into the plasma and then into the alveoli, where we can now exhale and get rid of that byproduct of respiration, and therefore we have just completed our respiratory cycle. We've loaded with oxygen, and now we've unloaded our CO2, and we're ready to do the whole thing all over again. Now, the last part of our talk is really going to involve this acid-base balance, because remember, the higher the CO2, the lower our pH. And our plasma is going to need to stay within a very narrow range. We're not going to deviate from that much at all. But one way we can affect our blood plasma are either by volatile acids or non-volatile acids. Carbonic acid, that's our chief volatile acid because we can convert that into a gas. But non-volatile acids such as lactic acid, fatty acids, ketone bodies, they're also going to have an impact on our blood pH, and we have to control that fairly carefully. But the great thing is bicarbonate and largely carbonic acid that's creating the bicarbonate. That can be a nice buffer, because the more hydrogen ions you make, that essentially you're going to form a lower pH. That can be bound up in carbonic acid. And even better, when we get to the next chapter on kidneys, if we have a low pH, which means a lot of excess hydrogens, we can eliminate that in our urine. And that's one of the reasons why our urine typically is going to be more acidic than the rest of our body. So here again, we've got our CO2, just a reminder. We form our bicarbonate, our chloride shift, but we're going to have hydrogen ions. And if we can't bind them all to hemoglobin, many of those are going to come out into our plasma, and that's going to affect our pH. So if we lower blood pH down to 7.3 or lower, that's not much, but it's enough to, cause us problems. Then we have acidosis. And if that acidosis is caused by hypoventilation, then we are going to have increased CO2, which is hypercapnia, and that results in respiratory acidosis. Do you see how the three relate to each other? And I cautioned you when we talked about CO2 before in a blood unit, pay attention to the question. Because if we talk about the disease or the clinical situation caused by hypoventilation and low pH, that's respiratory acidosis. If we talk about what term describes high carbon dioxide, well, that's hypercapnia. So those terms aren't interchangeable. They're not synonyms. They don't mean the same thing. So pay attention to those questions. But we can also have a situation where our blood pH is lower because of some metabolic problems. We have a rise in non-volatile acids. So if we have diabetes mellitus and we're not actively incorporating the glucose into the cells and we're breaking down protein, then we're going to have excess ketones. And that's going to lead to acidosis. Or in this case, our pancreas produces a lot of sodium bicarb to offset the acids that are present from our stomach coming into our intestines. But if we have diarrhea, then we're going to be flushing that bicarb out of our body before we can absorb it. So we will be losing a lot of bicarb and have too much acid, and so that results in metabolic acidosis. Now, flip it around. If we increase above 7.45, We have respiratory alkalosis. Hyperventilation now leads to hypocapnia, and that's our respiratory alkalosis. Metabolically, if we get too much bicarb from some clinical infusion, that's one thing. Or if we are nauseated and vomit, then we're going to be eliminating all of that hydrochloric acid from our stomach. Now we've got too much bicarb from the intestines that's being absorbed, not bound up or offset buffering the acid, and that's going to lead to an increase in pH of our blood and alkalosis. Fortunately, our kidneys are going to play an important role in balancing out pH as well. This is not simply a factor of respiratory or the metabolic systems. For your studies, here's a table that shows you plasma CO2, normal, low, high, what happens with certain conditions of metabolic or respiratory, and the causes of those. And then here we also see, relative to ventilation, how we can have respiratory acidosis, alkalosis, or this metabolic acidosis, where our pH is low, CO2 is low, which will lead to hyperventilation. But that's going to be a compensatory effect because of metabolically what's going on. And with your CO2 low, you're going to be trying to hyperventilate to offset that.